Okay, I think we should start now. Yeah. So I'm very pleased to host and chair today this outstanding webinar that has been organized by the FENS Committee for Higher Education and Training. Today's webinar is entitled Periphery CNS Miscommunication in Neurodegenerative Disorders, Focus on Brain Barrier Pathology. And I'm very pleased to welcome eminent scientists in this field, such as Professor Britta Engelhardt. She is a professor of immunobiology and director of the Theodor Kocher Institute of the University of Bern. And she will give us an overview of brain barriers and immune trafficking across brain barriers. Then we will listen to Jonathan Kipnis. He is a distinguished professor of pathology and immunology and director of the Brain Immunology and Glia Center of the Washington University in St. Louis. And Professor Kipnis will draw our attention on less investigated barriers. And he will show us the latest research in meningeal lymphatics, the lymphatics system, and even skull bone marrow. So we listen something extremely exciting and new from Jonathan Kipnis. And finally, we will listen to Professor Gabriela Constantin, which is a professor of pathology and immunology and head of the laboratory of neuroimmunology and neuroinflammation at the University of Verona. And she's an expert in trafficking of immune cells through the brain barriers. So she will tell us about the most recent research of immune cell interaction and trafficking in the meninges during neurodegenerative diseases. So the first speaker will be Britta Engelhardt. And so I will leave the stage to Britta. Britta, thank you.
Thank you very much for this kind introduction, and I welcome everybody here to the first part of the seminar. So what I will do is, in principle, to show you how we can image the brain barriers and introduce the brain barriers. Which barriers are we talking about? And the brain barriers maintain the CNS immune privilege. That's the theme of the introduction. So when we look at the brain barriers, you have here a coronal section of a human brain. What everybody knows usually is the blood-brain barrier. And the blood-brain barrier is localized at the microvessels in the central nervous system. And it's at the level of the capillaries, but also of the post-capillary venules. These endothelial cells are very unique. But if you look very carefully, you see that these endothelial cells are here embedded in a basement membrane. This is depicted here in yellow, high number of pericytes. But then there is a second basement membrane, which is referred to together with the astrocytes that embrace the vessels with their end feet to the glia limitans. So the central nervous system has two barriers, so to speak, one at the level of the endothelial cells, and then there is a second barrier towards the parenchyma where the neurons reside. Now, I will not talk about the choroid plexus today. The choroid plexus you find in the ventricles of the central nervous system. The stroma folding out from the meninges has fenestrated vessels, and the barrier towards the central nervous system is made by these epithelial cells that form a blood cerebrospinal fluid barrier between the stroma and the CSF-filled fluid space. Now, the other barriers, which we will focus on a lot today, are on the surface of the brain and spinal cord. And they have a multi-layered setup. So from the outside, if this white area here was the bone, so to speak, in the brain, then below the bone, as a periost, so to speak, you find the dura mater. The dura mater has its own blood supply that is different from the brain, and it also harbors lymphatic vessels. Below, it's separated because it doesn't have a blood-brain barrier by the arachnoid barrier. This is a multi-layered fibroblast sheet from the subarachnoid space. And the subarachnoid space, again, contains cerebrospinal fluid. It's connected here by these trabecula towards the lower layer. This is the pia mater. This is the third layer here that does not really form a true barrier. I'll show you later. It does for cells. There is collagen below, so you have a subpial space. And then you can appreciate that to the surface, we again have the glia limitans. So there is a second barrier. So also on the surface, if you look for the blood compartment, you have the outer barrier and an inner barrier towards the parenchyma. And all these different barriers are made by endothelial cells, epithelial cells, glial cells, and then meanwhile, we also know by fibroblasts, and they ensure CNS immune privilege because they divide the central nervous system in different compartments. Now, the misconcept about the immune privilege, to just quickly introduce this to you, came from physiology and pharmacology, because at the time when these people studied the blood-brain barrier, they found that there is no free diffusion of molecules from the blood into the central nervous system, and this is due to complex and continuous tight junctions. In addition, these endothelial cells of the blood-brain barrier have almost no pinocytic vesicles, meaning there's also no free diffusion of molecules through the endothelial cells. However, everything that needs to get transported in or transported out of the CNS is done by specific transport processes. So the immunologists at the time thought, well, something like a T cell would not be able to cross this barrier. And this brought the misconcept of immune privilege as immune ignorance into the field for a while, where people thought that the immune system does not have any access to the central nervous system. Now, in the meanwhile, we know that this is definitely not true. Many research groups, including ours, have shown that immune cells can cross in the absence of neuroinflammation at the level of the postcapillary venules of the blood-brain barrier. They can cross veins and enter into the subarachnoid space or the surface of the brain. And T cells can also use the choroid plexus as entry site to reach the brain. So there's 3 potential routes of T cell entry into the CNS. Now, this always has brought about now vivid discussion that there is no immune privilege of the central nervous system, and that this is history. So there are many articles written like that. And as we have here also an educational talk, I would like to show you the way how we discuss in my lab always the immune privilege, because we say it's a historic concept. And we base this on a castle that's sitting on a hill. So this is actually the Marienfestung in Würzburg in Germany, where I was educated with Hartmut Weckele. So I have very fond bonds to this city. And if you look very carefully, you see the architecture of the CNS immune privilege right there. So you have a castle, and that's surrounded by 2 walls. Now, the outer wall, if we stay at the level of the blood-brain barrier, would be our blood-brain barrier, and the inner wall is the glia limitans. Remember, no other organ has 2 barriers towards the bloodstream and the periphery. In the castle, so to speak, so the CNS parenchyma, the neurons would communicate and have a homeostatic environment. Then we also have a true castle moat. This would be our perivascular subarachnoid spaces, which are filled with cerebrospinal fluid. Then we have the antigen-presenting cells, or the castle moat guard, so to speak. And in case of danger in the periphery, activated T cells can cross the outer barrier and interact with these antigen-presenting cells in order to see if they see their cognate antigen. And if they do, then it would be the signal that there is a danger in the central nervous system. They would proliferate. Now you have opening of the doors, so to speak, of the outer barrier, the blood-brain barrier. In the real world, this means trafficking molecules are upregulated and expressed, allowing more cells to enter. And eventually, these immune cells will cross the glia limitans and reach the parenchyma. And this is usually associated with clinical disease. Now, with this concept, if we now look at these brain barriers, how can we investigate this? So this would require that we can image and visualize these barriers together with the immune cells in the CNS. Now, I'm always stealing here a wonderful video from Alexander Flügel's lab where they show T cell extravasation from lymphomeningeal blood vessels. If we now look at the cell that we're following here, you can see that eventually it has reached the outside of the vessel now, but it's stuck here. So there is something that prohibits its migration, but we cannot say what it is, because we don't know, if we now look at this cartoon anymore, if this cell sits somewhere in the subarachnoid space and bangs its head, so to speak, against the arachnoid barrier, or has it crawled below the pia mater and is blocked here by the glia limitans, or has it actually reached the parenchyma and it's just such a dense environment that the cell is stuck? Or has it extravasated up to the dura and is stuck in the dense fibroblastic tissue? So we don't know. So what we need is, in principle, reporter mice allowing us for in vivo imaging where we see the barriers. And we and others have used 2-photon intravital microscopy that allow you in a confocal technique to make movie stacks and follow immune cells in the tissues over time. And we have started to use a reporter mouse that's called the E-cadherin GFP. It's a knock-in mouse of the adherence junction molecule coupled to GFP in the endogenous locus. And what you can appreciate is that you see here the vascular junctions. And we have used this mouse because before in vitro, we had found that T cells in the blood-brain barrier cross mainly via these tricellular junctions in order to extravasate. So this was the original goal, to look at this. Now, when Josephine Mapunda was starting to make a spinal cord window and doing 2-photon imaging in order to look at the cells, this is what she found. And I think you can appreciate, this is where you see the red color. This is the vasculature. So we have seen the dorsal vein, but you can appreciate that beyond the vascular junctions, you see here way more junctions. The second harmonic generation in blue showed you the dura mater, and then below, I think you can see there are several layers of tissue right below the dura and right below the vasculature at the expected locations of the arachnoid and pia mater. And you can really appreciate the junctional connections. So obviously the E-cadherin GFP identifies spinal cord leptomeningeal layers. And we have also seen this for the brain. Now, if we look at this, so at the expected level of the arachnoid, you can see these junctional connections. So this is right below the second harmonic generation of the dura mater. And then if you go further down, you see at the level of the pia mater, you can also see beautifully these large cells connected by the E-cadherin GFP positive junctions. If you then turn the data set and make a YZ MIP, maximal intensity projection, you can appreciate this is the dorsal vein, this is the vasculature, you see here the arachnoid mater, the dura mater, you see a subarachnoid space in the trabeculae, and then the pia mater. So then, of course, we were interested, where are these cells exactly? So what was known is that E-cadherin is expressed at the arachnoid barrier. So this is the outer layer of the arachnoid. And if you just follow me here, looking at the blue and the red boxed areas here in a higher magnification, this is optical sections, you can nicely appreciate that here the arachnoid barrier in magenta, these are the E-cadherin positive cells, and the E-cadherin cells are just below, maybe a little bit overlapping, but it's not the arachnoid barrier, it's the arachnoid cells right below, and you can appreciate this here once more without the nuclei. Then you may have seen that there was a report, there is an additional layer in the subarachnoid space, which I've not talked about, and this would be cells expressing PROX1. So when we crossed our E-cadherin GFP reporter with the PROX1 reporter, and again, we can just look here at the lower area with the magnification, you can appreciate that the E-cadherin, so this is again our barrier cells, is exactly beside the PROX1 positive cells. And if you follow me over here, you can appreciate again that the PROX1 positive cells are embedded in the E-cadherin GFP positive arachnoid fibroblasts. So this cell layer is an integral part of the arachnoid mater. Now, just to summarize, we were not the first observing this, but if we look at the seminal work of Key and Retzius from 1875, they have already looked at the arachnoid and the pia, in dogs and rabbits, as you see. But I think you can appreciate that what they were finding with silver stains in the junctional patterns, that we have now been able to localize a molecular cue right into these junctions, which are made by the E-cadherin. So can we now image the CNS immune privilege as I've promised you? So what we did, we looked at an animal model for multiple sclerosis. This is experimental autoimmune encephalomyelitis. You can read this here. These animals get a clinical disease. You see here they are paraplegic. And what happens is that immune cells depicted here in red cross the postcapillary venules. And as I told you before, when these immune cells reach the parenchyma, this is the time when these animals develop a disease. So using this model, we just introduced EAE in the E-cadherin GFP mice. And when we now looked at this, this was the subarachnoid space in the healthy mice, in the E-cadherin GFP reporter mouse. And I think you can appreciate that now we can see something we were not able to image before, that the subarachnoid space in the spinal cord widens massively. And here you can appreciate the beautiful trabeculae. So there's a change in cerebrospinal fluid compartments in EAE that has not been appreciated. Then, of course, we looked at the injection of tracers, and so in order to investigate if there is true barriers formed. And I just give you here an example. So when we do this, you can see that the tracer arrives. You can also appreciate maybe that some of the macrophages sitting there, so the castle-node darts, are eating these molecules. So what you can appreciate, and this is a BSA, that both in principle in the healthy mice, that the arachnoid and the pia really provide a barrier. We can also look at T-cell interactions now. And what you will see here on the left, this is the top view, so to speak, and on the right side, you will see the side view. And what we're looking at here is CD8 T-cell trafficking in a healthy mouse. And these T-cells have actually reached the subarachnoid space and are crawling, what you can appreciate here, above the pia. So we were very excited when we observed that, because that means that these T-cells recognize the pia as a barrier. They can eventually also cross, which they do only in very low numbers in healthy mice, where this is in principle only for the part of the immune surveillance. We can also go back and look at the pathway these T cells take crossing the pia mater, which will be shown in the follow-up videos. We found, as for the endothelial cells, that they can cross the junctions. So this is called paracellular diabetes. But we have also observed T cells crossing from above to below the pia, away from the junction, so they may pour in these PL fibroblasts. And then last but not least, we were of course also interested, and this will be the last segment of this video, if the T cells can escape, because this is still an unanswered question. What do T cells do that have gone to the subarachnoid space and do immune surveillance? And so far in the spinal cord, we have only found this one single cell that's poking here heavily into the arachnoid, but it doesn't leave. So I hope I could convince you with this introduction that having suitable reporters, and with the Egalitarian GFP reporter, we can now see, so to speak, the roof and the floor of the subarachnoid space. And this allows us to localize where the immune mediators and the immune cells are located, and can distinguish between immune surveillance and immune infiltration and neuroinflammation. So I hope I have convinced you that these brain barriers maintain CNS homeostasis and establish the CNS immune privilege by zoning the CNS in compartments that have different accessibility for immune mediators and immune cells. I would like to thank the fantastic team. So part of this was the PhD thesis of Josephine Mapunda and Javier Pareja. And as you see, she is from Tanzania. We celebrated her PhD defense. I have to thank the fantastic team. And I have to thank all the collaborators and the funding agencies and you for your attention. Thank you very much.
Thank you very much, Britta, for this beautiful presentation. And before we go ahead with the next presentation, I need to remind, now all the participants have joined the meeting, so I needed to remind that if you have any question, please write it in the Q&A session that you will find at the bottom of your screen, because the chat is closed. And please sign your questions, because anonymous questions will not be considered. Thank you. So now is the turn for Professor Jonathan Kipnis, which will talk us about immune surveillance at the brain borders. John, please.
Thank you very much, Anna, and thank you for inviting me here, and welcome to the audience. I was telling my co-panelists that yesterday was a big holiday in the US, Thanksgiving, and so we had here lots of people until about 2 a.m. and it's now only 8 a.m. So if I'm a little bit off, then you guys know why. So let me tell you, so because it's a Zoom meeting, we decided that we will only present the published data, unfortunately, so I apologize there. This is very quickly my conflict of interest, and let me start. So Britta gave a wonderful introduction, so I don't need to go deeper into it. Just to kind of very quickly summarize, so we have the meningeal compartment right here with the pia, arachnoid, and dura touching the skull. And then underneath is arachnoid, and then pia, and the CSF is flowing between the arachnoid and the pia mater. And what's happening in this, although within the brain, our main immune cell is probably microglia, even though we're now getting more and more very interesting findings are coming out from different labs that maybe there are other peripheral immune cells sitting within the brain. Their numbers are quite small. And but if we're looking in the dura, in the meningeal compartment, mainly in the dura, then we will see a great variety of immune cells, and they produce different molecules, cytokines, chemokines, and these molecules are getting access to the brain through those barriers that Britta so nicely discussed. And they actually have effects on the brain. There are we were focusing on a couple of them from CD4 cells. There are now many other labs that are showing also these really interesting effects. What else is found here in this meningeal compartment? Again, mostly in the dura, lots of immune cells. Also in the dura, we have lymphatic vessels. And so, for example, this is a whole mount of the dura mater. And here is labeling for CD31 is a marker for endothelial cells. So these thick vessels are the sinuses, it's the venous structures that are draining all the blood from the brain. And if we're labeling with LIFE1, which is one of the markers for lymphatic endothelial cells, you will see that along these sinuses, we have structures which are vascular structures. And very important, LIFE1 is not sufficient to label for lymphatic endothelial cells. And so we're showing here that they express all the other molecules lymphatic endothelial cells are expressing. So these are real lymphatic vessels. So here is an interesting thing. Remember that what I showed you, lymphatic vessels, they are in the dura, which is outmost layer of the meninges. So why do we even care what's happening there when it's a peripheral, fully peripheral tissue? So what is happening is here, we're looking at the video in the mouse, and we're injecting this mouse with two different tracers in the CSF, with a 0.1 micron bead and with a 3 kilodalton dextran. And what you will see now is that these tracers, after about 6-7 minutes, are showing up in the dura. So there is a molecular passage is happening from the CSF into the dura. We actually just now have a new story, which hopefully will be out at some point, where we identified how exactly this molecular passage is happening. Once the material is in the dura, then it gets, here you see in red, it gets into the lymphatic vessel. Okay, so from the brain into CSF, from CSF into the dura, from the dura into lymphatic vessels, and then from lymphatic vessels, it goes all the way through those lymphatic vessels, down the neck, and then it gets into this draining deep cervical lymph nodes in mice. This is a mouse anatomy. In humans, we have many more lymphatic vessels, so it's more complicated. But first of all, humans do have meningeal lymphatics as well. The structure is very similar to what we see in mice. And humans also have molecular passage of molecules from CSF into the dura mater. So we believe that what we're showing in the mice, this actually, let me summarize it here. It's right coming. Okay, and then what I forgot, what I didn't tell you yet, is about the lymphatic, right? So they showed you now with the CSF into dura, into lymphatics, into the lymph nodes. Now, how does the brain and CSF connect? We have a beautiful system described by Mike and Neurodegenerative now all close to 10 years ago. And what Mike showed is that CSF actually is moving through the brain. So CSF is produced. It is diving into the brain along the arteries which are going into the brain. Because of arterial pulsation, CSF will be pushed out and will become part of the interstitial fluid of the brain. And then will propagate to the brain tissue, which is still a very major question. How does CSF propagate through the brain tissue? Again, I think we have interesting answers, which hopefully will be out soon as well. And anyway, and then at some point, it will be recollected in the perivenous space. So if we summarize what I just told you, we have clean CSF, which gets into the brain through the lymphatic system and then comes out as dirty CSF. By dirty, I mean it carries now the molecules from the brain. It could be metabolites, which you want to cleanse, but it also has peptides or antigens, those that will be presented to the immune system. And then it gets into the dura mater. And from dura, it gets into the lymphatic system. Now, why is dura mater so important? Because as Britta very nicely told you, we have these layers of barriers. And it's very important, of course, for the brain to not have all this immune interaction happening directly within the brain tissue, because brain is a very unique and very sensitive organ. We don't want immune cells crawling through it. So the brain developed throughout evolution the system where it pushed all of its immune activity, immune surveillance to its borders. So if we're looking into that border that Britta showed you very nicely just now, what you see is that on the sinus and also elsewhere, you have lots of macrophages, and many of them express very high levels of MHC class II, which is a molecule that is required for a T cell to interact with the macrophage and for recognition of the peptides. And here in yellow, you will see these are T cells. So if we're looking in a healthy mouse, there is lots of T cells in the dura. And funny enough, they all concentrate around around the sinus. So what we think is happening is the CSF is bringing materials from the brain through, in humans, arachnoid granulations. In mice, it's probably happening through some other molecular passages. Macrophages will uptake these molecules and will present them on their MHC class II. And the T cells can very easily cross the sinus because it's a very large vessel, very slow blood flow, lots of adhesion molecules, and lots of chemokines. So all the wonderful environment for T cell to be able to cross. And then T cell will speak with its macrophage and will sample the antigen, right? So because remember, brain is a very important organ. We don't want immune cells to go inside the brain, but we still want to make sure that brain has immune surveillance, because if there is an infection in the brain, we still want to send the army, the immune cells, to clear the brain. So again, instead of going in the brain, we believe that this interaction happening in the dura. And then if there is no recognition, so there's no problem, then this immune cells, T cell, will leave through nearby lymphatics. If there is interaction, that immune cell will start proliferate, will start the inflammation, but the real pathology will start when the cell will go dive into the leptomeninges and, of course, farther into the brain tissue itself. So when the barriers will be actually breached. So now, but how now is the problem? So if molecules from CNS are presented to immune system and professional antigen-presenting cells and are draining to lymph nodes via lymphatic routes, then what makes CNS immune privileged? Because if we're going by the old definition, the definition was that immune cells can't cross the blood-brain barrier, which is not true. And the second thing is that the brain has no lymphatic vessels, which is only partially true, but there is lymphatic drainage. So here I'm telling you, we still have immune presentation on APC. We have immune presentation in the draining lymph nodes, yet the brain is a very, very immunologically unique organ, right? So if we're going back to Medawar's or even before to his predecessors' works, if you transplant a piece of tissue or tumors into the brain, they will survive there very, very well, as opposed to the periphery. So immune cells can't very efficiently reach them in the brain. So what is happening? And we thought maybe immune system in the brain border is very special, right? Actually, the whole story started with a student in my lab, Andrea Kubura, who is now back to Italy and is doing postdoc with Matteo Nicola. So what Andre realizes, if you put two mice in parabiosis, so they will now start sharing blood. One is a GFP and one is a normal mouse, a wild-type mouse. And if you look then into immune cells and T cells into this gray mouse, you don't see much difference. But if you look into monocytes or neutrophil, you will see that while this black mouse or normal mouse, wild-type mouse, has about 30% of green monocytes in the blood, in the tissue, in the dura, it only has about less than 10%. So why is it there is no good replacement of cells? Because normally monocytes are coming from the blood. And of course, we realized, well, it is very possible that the cells are being provided constantly by the skull bone marrow. Skull is a flat bone. And several years ago, groups of Matthias Nerendorf and Dorothy Sipkins showed that there are these unique structures, the dural skull channels. And these channels actually are delivering what they show, delivering cells during pathologies. And we said, okay, maybe they also functioning during physiology. So we actually took the mouse, we took a piece of skull and put here transplantation of GFP skull. As you can see, the skull survives quite well. The cells are here. And now actually we can look for green cells because they can only come from this one piece of little piece of skull. And as a matter of fact, you can see those cells in the underlying dura. We focused on myeloid cells, and our colleague Marco Colonna, who was published back to back with us, was focusing on B cells. And today we know that many immune cells, B cells, maybe even some T cells, but definitely myeloid cells and neutrophils and monocytes are all coming from this skull bone marrow into the underlying dura and physiology. Now, if we induce EAE in this parabiotic couple, and we are looking into the now spinal cord itself, not the borders, but the tissue, what we will see is that about 2/3 of the cells are coming from local sources, and about 1/3 is coming from the blood. And we think, even though it's not really, we still need to prove it better. It seems that the cells that are coming from local sources are more anti-inflammatory, whereas cells coming from the periphery are more pro-inflammatory. But again, I think the jury is still out there. So we, by the way, just published, yesterday came out a very nice review written in collaboration with Matthias Nerendorf in Nature Neuroscience. And if you want to learn more about this brain, about the skull channels, you can find it there. So why then skull and vertebra bone marrow monocytes are different from blood monocytes? And what we actually found, really by serendipity, is that when we inject our tracers into the brain and they reach the CSF, and then they continue to fly up, when now going from the brain up, they actually are going through those channels all the way into the bone marrow, and now macrophages in the bone marrow are uptaking those tracers. So the CSF not only goes into the dura, but it also reaches the skull bone marrow, and of course can change the content, composition, and the behavior of cells there. So this is just one example when we can inject actually antibodies against progenitor cells. We put the antibodies in the CSF, and they reach all the way into the dura and then into the skull, and are labeling 100% of progenitors. So very, very high efficiency. So this is so we published our paper in 2000 last year. Matthias published a paper back to back with us, showing how bacteria can use the same route. And then this group from Norway of Riksted Eide, they actually show that again the same biology is working also in humans. I must say they published before us, so they published in humans, and then we show that same biology also works in mice. So it's really wonderful that all these new anatomical conceptions are actually shared between mice and humans. And a recent paper, beautiful, by Ali Erturk's lab, showed that again the skull bone marrow is very interesting and very unique, not only in mice but also in humans. So what we are thinking is that the brain is instructing this bone marrow in the skull, and also in vertebra, by sending messages with CSF. This could be a good thing, but also could be a bad thing. And maybe the cells that are coming down are coming with a very unique phenotype and are not attacking the brain as badly. So what I will summarize: the meninges serve as a neuroimmune interface, and meningeal immunity is tailored to CNS needs probably. Skull bone marrow supplies immune cells to brain borders, and CSF enters to and regulates skull bone marrow. And of course, this is the lab. I showed you works of several people. Many of them actually left already, and not even in this picture. Some of them still are. Many collaborators, funding sources, and thank you very much for your attention.
And thank you very much, John, for this beautiful and such new data for us. I know they are published, but for the whole concept, these are extremely new data that open really new scenario on the field. So now I will leave the stage to Professor Gabriela Constantin, that is sharing her screen. And she will tell us about immune cell interactions at brain barriers during neurodegenerative diseases. Please, Gabriela.
Okay, so hello everybody. Thank you very much, Anna, for inviting me to this webinar. So for this topic, my presentation will regard neutrophil migration as CNS barriers, and we'll see how the context matters a lot. So For those of you who are less familiar with neutrophils, these cells were classically associated with acute inflammation. However, in the last decade or so, it was shown that neutrophils may play also a role in chronic inflammatory diseases. So neutrophils can rapidly migrate from the blood vessels into the tissues, where they can release cytokines, they can degranulate and release many types of enzymes, they can produce reactive oxygen species, they can also release these very toxic structures called neutrophil extracellular traps, nets, and they can perform phagocytosis. And so in the first part of my presentation, I will talk about the role of neutrophils and Alzheimer's disease. And studies performed in our lab in collaboration with Bruno Bonetti from our university showed that neutrophils here in green, these green cells here, accumulate in the brain parenchyma of subjects with Alzheimer's disease compared to control. And you can observe here that neutrophils accumulate mainly in areas rich in amyloid beta deposits, shown here in red. And we also noticed that neutrophils can also adhere inside blood vessels, which also have amyloid beta deposits. And we also quantify the data. And as you can see here in this picture, in this graph, there is a clear increase in the number of neutrophils, both inside blood vessels and also inside the parenchyma in patients with Alzheimer's disease compared to controls. And when you look more carefully, we observed that in blood vessels, which also had amyloid beta deposit, there were more neutrophils, and looking carefully at their morphology, it looks like these cells are spreaded and also degranulated, so they are strongly activated. And we also noticed that there is presence of these structures called neutrophil extracellular traps that can be extremely toxic in the case of sterile inflammation. And you observe the presence of these structures not only in the parenchyma, but also inside blood vessels, suggesting that these structures may somehow damage the blood-brain barrier. So in order to better understand the role of neutrophils in Alzheimer's disease, we use two transgenic animal models of Alzheimer's disease that many of you are familiar with. And so the first one is the 5XFAD mouse, which develops very rapidly amyloid pathology. And then the second transgenic animal model is represented by triple transgenic mice, which develop both amyloid and tau pathologies. And in both these animal models, we notice an increase in the number of neutrophils, shown here in red, in the meninges, leptomeninges. We also observed an increase in the number of neutrophils adhered inside blood vessels, as I showed also in patients with Alzheimer's disease. We observed the presence of these cells at the level of choroid plexuses and also inside the hippocampus. And when we quantify the number of neutrophils accumulated inside the brain, so able to cross the blood-brain barrier, we notice clearly an increase of the number of neutrophils at different time points of disease. And interestingly, we observed a peak of neutrophil infiltration, which coincides with the memory deficit, with the beginning of the memory deficit in this mice, suggesting that neutrophils may have a negative role, so a role in the pathogenesis of the disease. So indeed, when we temporarily depleted neutrophils from these mice by injecting an anti-LY6G antibody, we notice an improvement of cognitive functions in mice with neutrophil depletion compared to animals treated with an isotype control antibody. And we observe this improvement of cognition in both contextual fear condition tests, but also in Y-maze tests. Suggesting that neutrophils indeed contribute to the pathogenesis of disease. And we also have data showing that neutrophil depletion leads to a reduction of neuropathological changes, but for lack of time, I won't show this data. And then we also performed two-photon microscopy to better understand how neutrophils behave at brain barriers. And we perform two-photon microscopy at the subpile level, more or less at this level here, shown here with this rectangle. And these experiments were performed in 5XFAD mice with fluorescent neurons. And to label amyloid beta plaques, we injected methoxy-04, so the plaques were labeled in vivo in blue, whereas neutrophils were ex vivo administered, so there are these red cells, which, as you can see here, they interact with the blood vessel wall in vessels which have amyloid beta deposit, as shown here. So this suggests that amyloid beta somehow favors the interactions between neutrophils and the endothelial cells in parenchymal vessels. And we also ask whether integrins such as LFE1 integrin may contribute to the adhesion of neutrophils on the wall of the parenchymal blood vessels. And as you can see here, wild type neutrophils in blue can migrate inside the parenchyma, as shown here, whereas when you administer LFE1 deficient neutrophils in red, you can see that these cells stay confined inside the blood vessel. So this suggests that LFE1 is important in the interaction, that this interaction between neutrophils and blood vessel wall. And LFE1 is not only important on the adhesive interaction occurring inside blood vessel, but it's also important in the motility, in the migration of neutrophils outside the blood vessels. And as you can see here, neutrophils can nicely migrate into the tissue before the injection of an anti-LFE1 antibody. And now let's see what's happening when we inject the antibody. So as you can see, after the administration of anti-LFE1 antibody, neutrophils remain confined in the close proximity of blood vessels. So these cells need LFE1 in order to migrate into the tissue. So please keep in mind this data, because I'll go back to this and I'll show you data from EAE which are different comparing to the Alzheimer models. And we also asked, what about meninges? And Enrica Pietronigro in our laboratory performed all mount preparation, and she showed that there is presence of LY6G, so neutrophils at the level of dura mater, and by also labeling blood vessel, we also can observe that many of these cells are in close proximity to dural sinuses, as shown here. And we also quantify the data in the dura mater, and we can observe that there is a clear increase in the absolute number of neutrophils in triple transgenic mice compared to wild-type mice, and also the percentage of neutrophils from CD45 positive cells is also increased in triple transgenic mice compared to healthy animals. Now, I will change the context, and I will talk about neutrophils and their behavior at the meningeal level in experimental autoimmune encephalomyelitis, which is a model of multiple sclerosis and was introduced by Britta. And this is a very complicated picture showing that lymphocytes and also monocytes can cross different barriers that Britta showed you during EAE and multiple sclerosis. So they can migrate at the meningeal level, choroid plexus level, but also at the blood-brain barrier level. So we quantified the level, the migration of the infiltration of neutrophils in the spinal cord parenchyma, in the leptomeninges. Why spinal cord? Because in most EAE models, spinal cord is the main site of inflammation. And we observed, so here you can see the different clinical phases of the disease as measured by the clinical score in these animals. And we observed that in the spinal cord leptomeninges, there is a high infiltration of neutrophils in the preclinical phase of EAE, whereas in other phases of disease, there is infiltration of neutrophils both in the leptomeninges and parenchyma. So we focus on meninges, and we perform epifluorescence intravitreal microscopy. So Barbara Rossi performed these experiments in our lab. And these are pile vessels, and these are exogenously injected neutrophils. These white cells are neutrophils interacted with the blood vessel wall, and you can see this inflammatory slow rolling. And these phenomena, so the interactions between circulating leukocytes and the blood vessel wall are very important because they can lead also to vascular damage, as shown here in our two-photon microscopy setting. In an EAE model. So here you can see a pile vessels, and all these black holes here are interactive circulating leukocytes which interact with blood vessel wall. And where you can see this red arrow, there is vascular leakage here. Now you can see it. So neutrophils, circulating neutrophils, but of course also other circulating leukocytes during EAE interact with blood vessel wall and can lead to vascular damage. And we also perform whole mount staining and observe that there is clear accumulation, presence of neutrophils at the level of leptomeninges. And in order to understand where these cells are coming from, we exogenously injected neutrophils, which were labeled, fluorescently labeled, and performed 2-photon microscopy. So these cells was obtained from mice, and they were injected intravenously. So you can see we can find these cells migrated outside the leptomeningeal vessels, as you can see here. And what we notice is that the cells are mainly confined in the proximity of blood vessels. And in order to understand which are the mechanisms keeping these cells in the proximity of blood vessel, we use an anti-LFE1 antibody. And as you can see, before the antibody administration, cells are mostly confined near blood vessels, whereas after anti-LFE1 antibody administration, you can see that these cells can finally penetrate and migrate inside the tissue. And this is clearly different. There is a clear context difference between EAE and the Alzheimer model. So if you remember in the triple transgenic mice, I show you that an anti-LFE1 antibody will block the migration of neutrophils inside the parenchyma, whereas here, if we block LFE1, we can see that neutrophils can migrate and can penetrate deeper into the tissue. And we hypothesize that probably neutrophils are engaged in cell-cell interactions with other local cells, and we thought, of course, macrophages, because the leptomeninges are rich in macrophages, and we performed 2-photon microscopy like shown here, and we injected fluorescently labeled neutrophils into the mice, and we can see that these cells can migrate outside the leptomeningeal vessels, and these cells, so the blue cells, literally run to encounter, to establish interactions with the green cells, which are CX3, CR1 cells. And this suggests that neutrophils can establish cell-cell contact with these cells in the leptomeninges of mice with EAE, and also perform some wall-mount studies suggesting again that there are contacts between neutrophils in red and also meningeal macrophages shown here in green. So to conclude, our data suggests that neutrophils participate to the pathogenesis of neurodegenerative diseases, and then there is a neutrophil CNS miscommunication during neurodegenerative diseases. And our data suggests that neutrophils can contribute to the pathology of CNS barrier because they can migrate into the dura mater, as you can see here. As I show you in the Alzheimer models, they can migrate into leptomeninges. They can also migrate into the choroid plexuses, and they can also cross the blood-brain barrier. So at the end, I would like to thank all the people that work on this project in my group, and also, of course, to our funding agency. So thank you very much for your attention.
And thank you, Professor Constantin, for this exciting new data, which I think are really pivotal for defining new therapeutic targets in neuroinflammatory diseases. So obviously, we have several questions for all the speakers, and we don't have much time, so I will try to pick one question for each speaker, if I can. And let's see. One question is for Britta, and comes from Laura Flutter, and she says, how do you image the mice in vivo without damaging the meninges? For example, when implanting a cranial window, the dura mater will be damaged.
Yeah,
so it's a practical question, but I think it's important to clarify.
Very important. And actually, thank you very much, Laura. This is a very important point. And we are now having these reporter mice in the unique situation to be able for the first time to see the damage. Because before that, nobody had this opportunity. The interesting issue is it's much easier to make a cranial window if you have a skilled neurosurgeon. And this is how we do this. I have a special neurosurgeon in the laboratory. You do not harm the dura mater with the cranial window. But if you would like to go to our manuscript, we have a full paragraph on the issues we encountered with skull thinning. And we discussed this endlessly with the neurosurgeons, because with the skull thinning, interestingly, you create a small subdural space, meaning that the dural border cells somehow rupture and you get a little bit more fluid into this space between the dura mater and the arachnoid when we don't understand, and nobody could explain this. But this is something we have to keep in mind, that these spaces widen maybe pathologically a little bit. And I think if we know this, then we know the small artifacts or the larger artifacts we create, but you only see them if you have a landmark for these outer borders. Otherwise, you would not even see this.
Thank you very much, Britta. So now I have a question for John Kipnis. The question is from Matteo Vergassola, so it's an Italian researcher.
And I'm sorry, do you mind if I answer a question from Eva Maria? Because it's a very interesting question here. So I said I will, and I'll try to type the rest, the CSF passage through the brain. Yeah.
Yeah, actually, I signed this question and then I went to the next one. Yeah, of course. So the question is CSF passage through brain parenchyma. Does this mean that when CSF is collected from patients, it represents clean CSF and not dirty CSF? This is a question from Eva Maria Kramer-Albers.
Yeah, I think it's a wonderful question, because this is something that I've been asking for a long time myself. What are we sampling? So, but before then, the question is, does all CSF go through the CNS tissue, or is there CSF which some of it is really clean, or some? I mean, because we see in a CSF brain-derived proteins such as amyloid beta and tau and all this, that suggests that we are sampling, at least some of what we're sampling has gone through the brain. We also see lots of synaptic material there, etc. But I think it is still very, very interesting conceptually. Is there a separation between the clear CSF and the dirty CSF, and what percentage of the clear goes to the brain? And I don't think we have the answer. Maiken published recently a paper where she suggested that there may be some separation with layers, but I think we're still very, very early in being able to answer this question. And I think it's really important. But even beyond that, when you're sampling CSF from humans, you're taking it from the very bottom of spinal cord. And so does it represent well what's happening in the cranial CSF? And even though they are mixing, but I think the mixing is not as good as we are learning from textbooks. So these are great questions, and I hope that students in the audience will look into it and study it in their own labs. Thank you.
Thank you, John. So a question for Gabriela Constantin from Damien Levard. He says, wonderful 2-photon videos with the neutrophil extravasation. Do you know if these videos were taken at the meningeal level or deep in the brain parenchyma?
Well, thank you very much, Damien, for this question. It's a very important question. So from our data that I didn't have time to show you, neutrophils can really penetrate into the brain parenchyma. We have videos, 2-photon microscopy videos showing clearly that neutrophils can also go into the meninges, okay, with 2-photon microscopy. The images I show data from vessels that were inside the parenchyma. So usually we can go beneath the pile membrane about 70-100 microns. So of course, not so deep inside. As you maybe notice, we cannot see the neuronal bodies, but the images I show that they suggest that they are in the brain parenchyma. Whereas, as I mentioned, yes, we have also data showing that exogenous injected neutrophils can also arrive in the leptomeninges.
Thank you, Gabriela. And there are more questions. Well, for instance, for John Kipnis. Let's see. Oh, I'm missing the questions.
Maybe last one. There's at the very end, Matteo.
Okay. Yes, this is the question I was looking for. Is from Matteo Gianeselli, and he asks, do you think monocytes infiltrate the brain during Alzheimer's disease? And if so, do you think they come from the skull, bone marrow, or the peripheral one?
Yeah, again, a very good question, which we are asking ourselves the question all the time. So as you know, there was about maybe 6-7 years ago, there were two kind of groups of scientists one arguing that monocytes do go in, and the other group arguing that there is no peripheral monocytes getting into the brain. And they each use a little bit different techniques. And I think this field is still not fully settled. I think we can say for sure that probably there is not a massive infiltration, like not what Gabriela showed, for example, of neutrophils in the meninges. I don't think we see that amount of monocytes getting into the brain. That's not happening. Meninges, yes. Brain, probably not. And now the question is, why not? Why can't dural monocytes get into the brains of Alzheimer's mice when they do into the brains, for example, of EAE mice or mice with CNS injury? And we actually think that this is all dependent on those routes of entrance, which I haven't talked today about. And again, hopefully will be out soon. And we think we have some indications that these routes may actually be clogged themselves by amyloid beta. And so if they're clogged, then cells can't get through. This is a speculation, of course. But anyway, to give you a more practical answer, if we take a 5-XFAD mouse and we put a piece of green skull on top, we can see some green cells getting into the brain. But the numbers are very, very, very low.
Thank you, John. And I would like to pose an open question to all the speakers from Laura Bojarskaite from the Oslo University, because I think this is an important question. Do you see differences in CNS periphery communication between different brain states, such as wakefulness, sleep, anesthesia, movement, sensory stimulation?
So I can maybe give one element that I know from studies made on my lab, that sleep deprivation will somehow, we don't know why, will activate brain endothelial cells, which will upregulate adhesion molecules, which potentially may mediate adhesion of circulating leukocytes. So we know that, for instance, sleep deprivation leads to increase in cytokine production, which can activate endothelial cells, and this may in turn promote leukocyte adhesion. So this can be a comment, a connection, observation that we had from studies made some years ago in our laboratory.
Mm-hmm. Mm-hmm. Yeah. Laura, thank you for the question. I think this is very difficult because these 2-photon imaging studies take very long, and I think not everybody has awake mouse animal permits. In the brain, this is not so complicated. In the spinal cord, I think this is killer. We are working on this, and I think it's a very important point you're making. It starts with something even milder. We have discussed with many labs already positioning of the mice changes, things that you observe in the cerebrospinal fluid. We know about the anesthesia. I think we're very well aware of, vascular effects of different anesthesia protocols. So we're a little bit in this limitation, what works for 2-photon and what would be good for observation. And I think most of us are working on this, but you are in a prime position to help us as you're working on these things.
Yeah, I agree with Britta. I think this is a very important question, not only sleep, but also positioning of the mouse. But I think that we really need to remember one thing, right? So during sleep time, when we're sleeping, our immune system is more active, especially when we are about to. So those of us who have little kids all know that the temperature, the fever goes up before the bedtime. That's our immune system. Actually, there's a group of Jan Bohr, I think, and Tanya, I forgot her name. They're both in Germany, and they actually have beautiful work on how immune system becomes more active in humans actually during night. So that's something that is very interesting and very important. And Maiken showed that also the glymphatic, this perfusion through the brain, also gets more efficient during night. So when we sleep, our brains probably are being washed more efficiently. So definitely, I think lots of things are happening in during sleep time that during wakefulness, we're probably just keeping all this process quiet, not to disturb the brain while it's it's working to keep us to keep us going. And I think, and I know for a fact that not only my lab, but several other labs are about to release some very interesting work on sleep. There is, by the way, another, I think the paper is out now. It's a group of from UCSF, and they showed that mice with a mutation, the natural genes that make them naturally short sleepers. So these people who can sleep very little, very few hours and function very well. This gene in mice makes them more resistant to Alzheimer's disease. So they sleep less, yet they are not developing AD as badly. So basically, they have a high quality of sleep.
Very interesting. So maybe one last question, since I mentioned it before, a question from Matteo Vergassola, which asks, are no specific proteins like receptor or channel that can be a selective target in the monocytes derived from the skull bone marrow compared to other monocytes?
Yes, it's a very interesting question, and actually we were very lucky to we think we found one molecular handle, what we call, that is expressed on one of. But I think, Matteo, if you know to do a very basic RNA-seq analysis, you can go to Ali Arturk's data set, which he published recently, and look, and you may find some interesting molecules there by yourself. We publish data sets as well, but ours are much smaller. Ali's cell paper has a much greater data sets also from mouse and humans. I would highly recommend you going there and kind of looking at his data. We think we have a molecular handle, at least for cells coming from local sources, but we need to do, we're now making mice and all this, so it will take a few years until we have the answer.
And thank you, John. So if we have a few more minutes, there is a question for Gabriela from Aishwarya Atioli, who asks, what about the direct interaction of neutrophils and tau and amyloid beta leading to neuronal damage?
Yeah, so this Aishwarya is really a great question. So we already have data showing that amyloid beta oligomers are chemoattractive for neutrophils, and they are also, so amyloid beta is also able to directly induce rapid integrin-dependent adhesion in neutrophils through FPR1 receptor, which is a G protein coupled receptor. So we think that maybe amyloid beta exposed, for instance, at the level of blood vessels can trigger integrin-dependent adhesion by directly activating integrins, which come from a bent conformation to an intermediate and fully open conformation, which leads to integrin binding to its counterlinear on the endothelial cells. We don't know about tau. We are studying this in our laboratory, but we think that neutrophils can somehow contribute to neurotoxicity during Alzheimer's disease.
Thank you, Gabriela. So I think we reached the end of this extremely interesting webinar. And so first of all, I would like to thank all the speakers for their availability, for their beautiful presentations, and for being so kind with all the participants and the questions. And also, I just want to remind, just 1 second. Okay. So for people that now that you see how a webinar works, for people that have ideas on topics for new training or educational activities that are currently not covered by FENS and that you think it could be considered by FENS, please submit your ideas. You will find all the instructions on the FENS website. So please consider this opportunity for all the participants. So again, thank you very much to Gabriela Constantin, Britta Engelhardt, and Jonathan Kipnis for making possible this beautiful webinar. Thank you to everybody.
Thank you, Anna, very much as well. Thank you.
Thank you. everybody. Bye, everybody.
And thanks to the audience for wonderful questions. Thank you very much.
Thank you.